you. Now, please welcome our third speaker, Xiaoyi Sun. She's a postdoc researcher from Beijing Normal University. She will talk about connectome-based growth models reveal neurophysiological subtypes of subthreshold depression. Thank you.
Thank you for the introduction. Hello, everyone. My name is Xiaoyi Sun, and I'm a postdoc at Beijing Normal University. It is a great pleasure to be here today to share our recent research. The topic of my presentation is about exploring individual heterogeneity and neurophysiological subtypes in subthreshold depression using Connectome-based normative models. And, I have nothing to disclose. Major depressive disorder is one of the most severe psychiatric disorders worldwide, affecting over 300 million people. Our group has also conducted a series of studies on major depressive disorder. Meanwhile, nearly 3 times as many individuals are affected by subthreshold depression, which refers to individuals who experience clinically relevant depressive symptoms but do not meet the full diagnostic criteria for major depressive disorder. Studies have shown that individuals with STD are at a higher risk of developing major depressive disorder. STD is typically defined using self-report skills and DSM criteria for major depressive disorder. In particular, according to the DSM-5, STD participants often show at least 1 core MDD symptom, but less than 5 in total. Consequently, different combinations of symptoms result in clinical heterogeneity among STD participants. However, the neurobiological substrates underlying this clinical heterogeneity remain largely unclear. The resting state functional MRI and brain connectome provide important tools to investigate the neurobiological substrates underlying clinical symptoms in STD. For example, the functional connectome alterations in frontal areas deformate network. However, most of them focused on the group level alterations. Given the high clinical heterogeneity among STD participants we mentioned before, we think it is very important to move a step forward to study the individual differences in the functional connectome in STD. So in this study, we aim to address these 3 key questions. First, how do individual differences in functional connectome alterations manifest among STD individuals? Then can we identify different neurophysiological subtypes of STD based on individual functional connectome alterations, and whether these subtypes differ in clinical and cognitive performance, gene expression profiles, and treatment responses. Here is a simple summary of the dataset we used. More than 1,000 healthy participants were from 10 research centers of DIDA-MDD group, and around 200 STD participants were from Jinan University. We applied strict quality controls and standardized the pre-processing to all the imaging data. And based on this dataset, we first calculated the functional connectivity strengths for each brain region, and the side effects were corrected using COMBAT. Then, based on these FCS values, we estimated typical lifespan trajectories of FCS based on the normative model in healthy participants. Then, for each SD participant, the FCS value of each brain region was positioned on the normative model to estimate their individual deviations. Finally, based on the individual deviation patterns, we identified neurobiological STD subtypes and validated subtype differences. Here are our results. For the individual deviations among STD participants, we found about 17% of STD participants showed extreme deviations in at least 1 brain region. And then for each brain region, we calculated the percentage of participants with extreme deviations, and we found no more than 3.5% of participants extremely deviated from the normative range. This finding suggests that while alterations in FCS exist in most of STD participants, the specific brain regions vary remarkably across participants. Based on the individual deviation patterns, we clustered SDD participants into 2 subtypes, and the subtype 1 showed severe deviations with positive deviations in default mode, limbic, and subcortical areas, and negative deviations in the sensorimotor and attention areas, while the subtype 2 showed moderate deviations with a converse deviation pattern. Then we compared subtype differences in clinical and cognitive performance, and we found the subtype 1, which showed positive deviations in the default mode, had more severe symptoms in the suicide item, depersonalization and derealization item, and poor cognitive performance than subtype 2. Based on the gene expression data from Allen Human Brain Atlas, we found the gene associations with FCS deviations were found only in subtype 1, and the PRS genes were enriched in biological processes, cellular components, and molecular functions, particularly those involved in brain development and transsynaptic signaling. To validate the subtype differences in treatment responses, we also included a subsample who received 8-week bright light therapy. And bright light therapy is a simple, safe, and effective method for treating depression and subthreshold depression. And we found both subtypes showed clinical improvement and normalization of brain deviations after treatment, but the direction of brain remission differed between the 2 subtypes. This finding suggests that the 2 subtypes might have different treatment mechanisms. We also use a baseline individual deviation pattern to predict the treatment outcomes in each subtype, and we found the baseline individual deviation map can predict the HDR change rate after treatment for only subtype 1, but not for subtype 2. Finally, we also validated our findings under different analytical choices. For example, we used the different threshold in FCS calculations and in the definition of extreme deviations. And we also validated the influence of head motion on the results by controlling for mean FD in FCS and applying a stricter head motion threshold. And the side effects were accessed by estimating the normative model based on the hierarchical Bayesian regression. And we also validated the subtype stability using an independent follow-up group. And finally, we also performed a balanced resampling analysis to reduce the potential bias caused by uneven sample distribution in the normative model estimation. And we found our findings are very stable. In conclusion, we observed the remarkable heterogeneity in functional connectome deviations in SDD participants, inspiring the identification of 2 neurophysiological subtypes, which differed in imaging and clinical features, gene expression profiles, and treatment responses. These findings highlight the neurobiological underpinnings of the clinical diversity in SDD, in emphasizing the necessity for developing personalized interventions for this condition. Finally, I would like to thank my collaborators Ying Wang, Qian Tao, and Guanmao Chen, my supervisors Mingrui Xia and Yong He, and all the DIDA MDD group members for their support in data collection and helpful suggestions. That's all for my presentation. Thank you very much.
Thank you very much. This is a very wonderful talk. So we have time for a couple of questions. Now we have one on the app, or I combine 2 into 1, actually. So can you elaborate a bit more on the normative model method you used in your work? How did you control for side effect? And also, what's the time interval between before and post-treatment? Use the mic. Thanks.
When we calculated the functional connectivity strengths, the side effects were corrected using combat, and we also addressed the side effects using by estimating the normative model using hierarchical Bayesian regression. It is proposed to reduce the side effects when you estimate the normative model.
And what is the time interval between pre and post-treatment? What is the time interval between the pre and post-treatment?
So the time interval. Post.
The treatment, 8 weeks.
8 weeks?
Yeah.
Okay, thanks. I think do I have time for another one? Yeah. So how did you come to 2 subtypes? Is it predetermined, or did the data yield 2 distinct patterns? How did you come up with the 2 subtypes? Did you predetermine, like, I want 2 clusters, or it's like purely driven by the data?
So your normative variation came up with 2 subtypes. So how did you know it's 2? Because you predetermine it's 2, or it's data telling you it's 2?
We use the k-means to cluster the participants, and we use NB class package to determine the optimal number of clusters. It can calculate more than 20 index to codify the clustering quality from different aspects.
Thank you very much. Let's move on to the next speaker.